I'm Matthew Klein, Chief Development Officer at PTC Therapeutics, and I'm happy to have this opportunity to share with you an overview of PTC's innovative gene therapy platform. In today's presentation, I'll begin by providing an overview of PTC's gene therapy strategy and pipeline, and then provide an update on our lead gene therapy program for the treatment of Aedc Deficiency, a highly morbid and often fatal genetic neurotransmitter disorder. For those of you who don't know PTC, we are a global diversified rare disorders company focused on discovering, developing, and commercializing best-in-class treatments for patients with rare disorders. We have over 1,000 employees with commercial activities in over 50 countries. Our corporate headquarters are in New Jersey, where we also have our gene therapy manufacturing site. Our international headquarters is located in Dublin. Since our founding in 1998, we have been dedicated to using innovative science to help patients. Over the past 2 decades, we have built a set of innovative discovery platforms that has allowed us to have a strong and diverse pipeline across all stages of development. We are proud that our research engine drives the development of innovative products for patients with high unmet medical need, including 2 innovative approved therapies, Translarna and Evrysdi. Today's presentation, of course, will focus on our gene therapy platform. Our overall gene therapy strategy is based on targeted therapy. That is, to deliver smaller amounts of gene therapy product directly to the anatomic area key to a particular disease's pathology. There are several key advantages to this strategy. Local administration lowers systemic immunogenicity and exposure. 2, by directly accessing cells with low turnover in the anatomic area linked to a significant aspect of disease pathology, we ensure that the gene therapy product reaches its intended destination and provides durable effect, which has been demonstrated in our Aedc program. And 3rd, lower dose levels means lower manufacturing burden, which is a key consideration in any gene therapy development program. Targeted CNS delivery requires the use of stereotactical neurosurgical procedures, which are commonly used in placement of electrodes for deep brain stimulation and other procedures. Stereotactic surgical techniques allow for minimally invasive, precise, and accurate delivery of gene therapy product to the desired intracranial location. Preoperative imaging allows for planning of trajectories and provides a Google map for a safe route to the desired gene therapy infusion location. Intraoperative real-time MR guidance can also be used to ensure and confirm safe delivery of gene therapy product. Our gene therapy pipeline includes both late-stage clinical programs as well as a number of preclinical programs. Our lead indication is aromatic L-amino acid decarboxylase, or Aedc Deficiency, for which we have submitted an MAA for approval to the European Medicines Agency and are in the process of preparing a BLA submission to the US FDA. We also have preclinical programs in Friedreich ataxia and Angelman syndrome, as well as a number of other early-stage development programs in therapeutic areas that are amenable to our targeted therapy strategy. Our lead therapy program is for children with Aedc deficiency. And as I mentioned, our MAA for approval of this therapy is under review with the European Medicines Agency, and we expect an opinion in the 4th quarter of this year. Aedc deficiency is a well-defined monogenic disorder that arises from pathological variants in the dopa decarboxylase gene that encodes for the AADC enzyme. Deficiency of the enzyme leads to an inability to synthesize dopamine and serotonin. Without neuronal dopamine, patients suffer from loss of normal motor development, and in many cases have profound motor dysfunction, including hypotonia, hypokinesia, dystonia, and oculogyral crises, along with behavioral problems, autonomic dysfunction, and developmental delay. Overall, Aedc deficiency is highly morbid and life-shortening, and there are no current treatments available targeting the underlying cause of the disease. Signs and symptoms of Aedc deficiency can vary greatly. However, they generally appear in the 1st year of life, and in a majority of cases, children do not achieve common motor development milestones such as head lifting, sitting, standing, and walking. These profound motor defects also increase the risks of aspiration pneumonia, malnutrition, and require intensive supportive care. There are currently over 80 disease-causing variants with both homozygous and compound heterozygous mutations reported in patients with AEDC deficiency. While a clear association between the severity of AEDC deficiency and specific variants has not been established, these mutations share in common a significant impact on dopamine production and therefore associated motor defects. Our gene replacement product, L-adopagene exuparvovec, which I'll refer to as AEDC gene therapy for simplicity, is designed to replace the defective DDC gene, regardless of the underlying mutation. The gene therapy product has an AAV2 mediated delivery system, and in order to ensure that the gene and its gene product reach the right destination, the gene therapy product is infused directly into the putamen. And as mentioned earlier, we achieve this through a stereotactic surgical procedure. And as I mentioned earlier, this directed therapy, this direct delivery allows for lower doses of gene therapy product and assurance then that the newly synthesized AEDC enzyme can be most effective in the right anatomic location. The delivery of the gene therapy product is accomplished through the stereotactic surgical procedure. And again, just to emphasize that this precise delivery is really essential, not only for efficacy, not only to make sure that the dopamine is produced in the right place of the brain where it has to be, but also for safety. We're able to precisely and accurately map a route from outside of the skull directly into that putamen using both CT or MRI guiding imaging preoperatively. I really want to emphasize this point that the neurosurgical aspects of this therapy is obviously critical to the long-term success of the gene therapy administration. Our AEDC gene therapy development program has been ongoing for over a decade, and over that time, the gene therapy has been demonstrated to be safe and effective in increasing dopamine levels and demonstrating related improvement in motor function. Long-term follow-up in patients has demonstrated that improvements in dopamine production and motor function are durable, with benefits measured 5, 6, 7, and more years after administration. I'll now walk you through some of the data that we've collected from our clinical trials. The clinical experience with our AEDC gene therapy product has been gained through 3 clinical trials conducted in Taiwan that include 28 children. In all 3 trials, 1601, 010, and 011, pediatric patients with confirmed genetic and metabolic confirmation of AEDC deficiency were enrolled and treated with AEDC gene therapy product through the direct bilateral intraputamental infusion. For the first 2 studies, we have follow-up data out to at least 5 years showing meaningful and durable clinical effect. For the final study, 011, 8 of the 10 treated subjects have reached the 1-year follow-up time point, and those will be included in the data that I share. This slide provides a summary of the endpoints collected in all 3 trials. The clinical assessments were utilized to capture potential treatment benefits on different aspects of AEDC deficiency, including motor function, achievement of developmental milestones, cognitive function, and language skills. In addition, standard clinical trial safety assessments and viral shedding measurements were used in all studies to gauge safety. Here's a summary of the characteristics of the children enrolled in all 3 trials. There were 28 children treated, and the 26 included in the analysis because they've reached the time point of at least 1 year of follow-up. The mean age at the time of treatment was 4.1 years, and the equal numbers of boys and girls treated. All subjects had profound motor deficits with no motor milestone attainment prior to the gene therapy administration. In addition to the clinical endpoints assessed, evidence of gene transduction and dopamine production were assessed using PET scanning. To capture biochemical evidence of treatment, subjects underwent pre-treatment and post-treatment 18F-DOPA PET scan evaluation. 18F-DOPA can be converted to 18F dopamine and then taken up by the nerve terminals in the putamen. And this allows us to quantify changes in dopamine production in the putamen where the gene therapy product is infused. A key objective finding in all 3 clinical studies was that all subjects demonstrated a statistically significant increase in dopamine production at the 12-month time point. Importantly, on long-term follow-up, these increases in dopamine levels were sustained in those children who've been assessed at 2 and 5 years following treatment. These findings provide evidence of increased and sustained gene transduction effect. This slide summarizes changes in motor function over time in all treated subjects who have reached the 24-month time point. All subjects demonstrated an improvement in motor function with a mean improvement of over 95 points from baseline to 24 months. Importantly, we also see that motor function improvements were sustained out to 9 years in the earliest treated subjects, demonstrating that the treatment effect is durable, which of course is incredibly important for a single administration therapy for a chronic disease. This slide summarizes the impact of treatment on the specific motor milestone items in the PDMS-2 scale across all 3 studies. This particular analysis focuses on 4 milestones: head control, sitting, standing, and walking. On this slide, we show the proportion of subjects achieving these milestones prior to and at 1, 2, and 5 years following treatment. Given the severity of illness, no children were able to achieve any of the milestones prior to treatment. As shown here, 75% were ultimately able to achieve head control following treatment, and 2/3 were able to sit unassisted. Several subjects were able to stand with support and even walk. At this point, not all subjects have reached the 5-year time point, which is why we have very few data at that time. However, it's important to note that milestone acquisition continues to occur over time, and we see that over time, patients continue to achieve more complex motor milestones. In addition to overall motor function, we also assess communication improvements and other neurological changes. As you can see on this graph, over time, treated patients show gradual increases in the mean total language score on the Bayley-3, which includes both receptive and expressive communication subscales. This endpoint was collected in the 010 and 011 studies, and again provides further evidence of important impact of treatment. In addition to overall motor function and achieving major milestones, we also noted improvements in other aspects of the disease, symptomology, as well as morbidity. We have, following gene therapy administration, treated patients were reported to have decreased dystonia, decreased hypotonia, decrease in respiratory infections, increased weight gain, and decrease in frequency and duration of oculogyral crisis, all of the signs of clinical benefit. Given that a picture, or in this case, a movie, speaks louder than static data, I wanted to share a video from one of the subjects treated in the 010 study. This is the first subject treated at age 2 months. Here's the patient treated here's the patient at baseline, profoundly hypokinetic, with no ability to sit or crawl. Here's the subject at 1 year following therapy, with the ability to sit and crawl. And ultimately able to stand with assistance. I'll note that he's wearing a helmet to keep him from banging his head as he learns to move independently for the first time. You'll now see here at 2 years after gene therapy, with now having the ability to walk with assistance, which is truly a remarkable result. In addition to the clinical benefits reported in these studies, the gene therapy was also reported to be safe and well tolerated. There were no treatment-related serious adverse events, and the most common adverse events were pyrexia and dyskinesia, and the dyskinesia occurred typically 4 weeks after therapy and abated over time. In addition, there was no viral shedding detected in post-surgery blood or urine samples, and no apparent safety profiles observed between the 2 different dose levels that have been studied throughout the development. So it's really important to note that in addition to those profound improvements in motor function, achieving a milestone, language acquisition, decrease in infections, and having durable effect over time, We also had a very strong and favorable safety profile demonstrated in these studies. In the remaining few minutes, I want to highlight our gene therapy program for Friedreich ataxia, which we are working to advance into the clinic. Friedreich ataxia is the most common hereditary ataxia resulting from a mutation in the frataxin gene. Friedreich ataxia is a progressive neurological and neuromuscular disorder that is highly debilitating and life-shortening. A key aspect of disease pathology is cerebellar dysfunction, which results in ataxia. And this occurs from frataxin deficiency, leading to a loss of cerebellar volume and function. Therefore, our gene therapy approach is to provide the frataxin gene directly to the dentate nucleus of the cerebellum. Again, we're delivering the gene therapy replacement directly to an anatomic location key to disease pathology. We are relying again on stereotactic surgical procedures for this targeted administration to the dentate nucleus. We've done extensive preclinical and non-clinical farm tox studies with promising results. On this slide, we demonstrate results from our preclinical studies in a porcine and non-human primate model of disease. Following targeted delivery of our frataxin gene therapy replacement to the dentate nucleus, we're able to demonstrate significant increases in human frataxin expression beyond the understood level of frataxin in the brain of unaffected humans, which gives us obviously the potential to correct the frataxin deficiency in people affected by Friedreich ataxia. We are advancing this program through the final preclinical stages and plan to be in the clinic in the near future. Thank you very much for this opportunity to share our experience in developing novel gene therapies delivered through a CNS targeted delivery approach to address serious unmet medical needs.